sometimes how would they ask the question is they will show you this graph, okay, and this graph is quite important. So to look at this particular graph here, this graph will explain to us the changes in the membrane potential of the axon over a period of time. What do I mean by that? So we are just taking one part of the axon, by the way. We are not taking the entire axon, okay? Now and the y-axis of the graph is the membrane potential and the x-axis is time, and I'm just drawing out, as you can see here, the purple color dot at the left is the sodium potassium pump, the orange color one in the middle is the voltage gated sodium ion channel, and the green color one on the right is the voltage gated potassium ion channel. At first, can this axon send an impulse? No, this axon cannot send an impulse, or it cannot generate the action potential. Why not? Because it doesn't have a potential difference, okay? It needs to first create and maintain a resting membrane potential. So how does it create and maintain the resting membrane potential? It uses ATP to power the pump, and the pump will transport out 3 sodium ions and transport in 2 potassium ions continuously until it makes the voltage inside lower and the voltage outside higher to negative 70 millivolts. This is referred to as the resting membrane potential. So that's still normal. But when a stimulus is given, it causes as you can see there, the voltage gated sodium ion channel opens. So what happens in this case over here, sodium ions rush in from a higher concentration outside to lower concentration inside. Thus, it causes the membrane potential inside to become more positive because it's getting more sodium ions, right? So when it gets more sodium ions, in this case, it becomes more positive inside compared to outside, so therefore the charge flips, okay? And this is depolarization, by the way. So at positive 30 millivolts, voltage gated sodium ion channels close and voltage gated potassium ion channels open. So in this case, go back over here to the resting membrane potential and just circling it there. Because we actively transported potassium ions in, the inside had a higher concentration of potassium ions, and also it had a lower concentration of potassium ions outside. So during this point, let's focus here. At positive 30 millivolts, the voltage gated potassium ion channels open. They only open at positive 30, by the way. So potassium ion rushes out. Therefore, the inside now is losing positive ions. The outside is becoming more positive. That's why as the inside loses more positive ions, they are losing the ions, right? They become more negative. That's why the membrane potential goes down again. So in this moment over here, the outside becomes more positive. Okay, this is repolarization. So as you can see, as potassium ion rushes out, okay, the cell is losing more positive ions, so the inside becomes more negative. It veers back into its negative boundaries, and therefore it reaches about negative 80, negative 78 to negative 80 millivolts. Okay, so at this moment of time, when it reaches here, the voltage gated potassium ion channels will close. From point number 3 to point number 4 is repolarization. That's basically what happens. But as you can see here, there's a pink color line over here. That pink color line is just basically time taken to return back to the resting membrane potential. And that period is known as the refractory period. For now, all you have to know about the refractory period is the axon is unresponsive to any stimulus, which means to say, if you were to give a stimulus, the axon will not generate an action potential. So as you can see here, the part where I'm circling here, that's the action potential, by the way. Depolarization and repolarization. This is the signal, okay, that is being sent along the axon.